Let's explore what meta-analysis is and why it matters for brain research. Meta-analysis brings together data from multiple studies through statistical methods to reach solid, reliable conclusions. Traditionally, this means sifting through publications, applying strict criteria, building a dataset, running analyses, and finally uncovering meaningful trends. What once took months of painstaking work can now be done in under 30 seconds with Neurosynth. A recent epilepsy study shows exactly how meta-analysis works in practice. Chen and colleagues examined how well AI and machine learning help diagnose and manage epilepsy, sharing their findings as an open preprint. Their AI models tackled 4 prediction tasks: identifying epilepsy presence, determining which brain hemisphere is affected, pinpointing lesion locations, and forecasting surgical outcomes. The team searched major databases, identified over 3,000 papers, screened them against specific criteria, and analyzed the 127 studies that made the cut. The results were impressive. Diagnosis hit 88% accuracy, lateralization reached 90%, localization achieved 82%, and prognosis prediction landed at 83%. This flowchart traces the journey from 3,227 initial studies through systematic filtering stages down to the final 127 included in the meta-analysis. Despite AI's promising accuracy, the authors found troubling bias in how studies recruited participants and conducted their analyses. Now let's turn to Neurosynth, the platform that makes brain meta-analysis remarkably accessible. Neurosynth combines machine learning, text mining, and meta-analysis to map relationships between brain activity and cognitive states. Forward inference predicts brain patterns from cognitive terms, while reverse inference works backward, identifying likely cognitive states from brain activity. This diagram shows Neurosynth's workflow. Searching for pain generates a meta-analytical brain map, then either predicts activation from terms or classifies patterns into cognitive states with probability scores. In the Tavakol study, Neurosynth synthesized 77 studies with nearly 4,000 activation points into a single cortical map. The resulting brain map highlights key regions, and the correlation plot shows how these patterns relate to actual behavioral measurements. Here's how to run your own meta-analysis on Neurosynth in 5 simple steps. Visit neurosynth.org, click Meta-analysis, select terms, type your search query, and pick your result from the list. Searching memory instantly processes 2,744 studies and displays activation patterns on a standard brain template, with clusters lighting up in the medial temporal lobes. The interface shows 3 brain views with red activation zones, crosshairs marking coordinates, and controls to adjust thresholds and visualization settings. Neurosynth also works in reverse, extracting cognitive terms from brain maps you upload yourself. Vos DeWyle and colleagues use this decoding feature to reveal what structural patterns in the temporal lobe tell us about cognitive function. Each gradient shown here corresponds to a word cloud below, demonstrating how brain organization reflects variations in sensory and cognitive processes. The brain's complexity resembles a wilderness full of unexpected patterns that neuroscientists work to understand. Meta-analysis pulls together data from many sources, organizes it systematically, and applies rigorous statistics to reveal meaningful insights. Tools like Neurosynth make it easy to generate these insights, opening the door for more researchers to contribute to our understanding of the brain. Now that you know the basics, you can take part in this endeavor by conducting your own personalized neuroscientific syntheses.